We are Group 1 from First DC Biotechnology, presenting the topic Isolation and Identification of Soil Microbes, and we have been appointed the research work, that is, isolation and identification of plant growth promoting rhizobacteria from cucumber rhizosphere and their effect on plant growth promotion and disease suppression. Our group members are Anna Maria, Bridget Johnson, Steffi Ausep, Viva Maria, and Swati Shankar. The authors are Sheikhul Islam, Abdul Akinda, Ananya Proa, MDT Islam, and MD M Hussain.
Introduction. The cucumber is one of the most widely grown vegetable crop in the world. The crop is prone to massive attack by Phytophthora capsule, that is crown root and blight. This plant pathogen infects host throughout the growing seasons at any growing stage and cause yield losses as high as 100%. Although fungicides can control the disease, their use is not good for the surrounding environment and to the survival of beneficial rhizosphere microbes. Plant growth promoting rhizobacteria are the rhizosphere bacteria that can promote plant growth and also suppress plant diseases. These microorganisms are able to enhance the recycling of plant nutrients and also decrease the use of chemical fertilizers. The objective of this study was to identify and characterize plant promoting rhizobacteria indigenous to cucumber rhizosphere and to evaluate their ability to suppress Phytophthora crown root in cucumber.
For the methods, we have included 5 steps of isolation and identification of soil microbes. First, the cucumber roots are collected, then it is sterilized, then done with the cereal dilution, and at last it is temporarily preserved. As I highlighted the term mortar and pestle in the third step. Mortar is a bowl typically made of hard wood, stone, etc. The pestle is a heavy and blunt club shaped object. It is used since ancient times. Here it is clearly showing the following steps of isolation and maintenance of rhizobium. Future perspective. It is suggested that only 17% of the known fungi can be readily grown in culture, as traditional methods have many pitfalls. Culture independent methods show great potential, and the perspective is effect of biocontrol agents Pseudomonas fluorescens 2p24 on soil fungal community in cucumber rhizosphere using TRFLP and DGGE. And if you don't know what is TRFLP and DGGE, it is clearly mentioned with the red arrow marks in the slide. Result and discussion.
Result: 10 isolates were identified on the basis of the ability to produce indole acetic acid and fixed nitrogen. It is also found that the bacteria produced more concentration of indole acetic acid in the presence of tryptophan. The selected PGR, that is plant growth regulator, showed consistent suppression of Phytophthora crown root. Phytophthora capsicum is a common fungal pathogen that causes severe diseases in plants. The result also suggests that the rhizobacterial treatment could also improve the germination and vigor index of the cucumber seeds. Vigor index is the sum of the root length and the shoot length multiplied with seed germination percentage. The total nitrogen content were also found to be increased in roots and shoots of the plant, and all the tested isolates were successfully colonized the roots of cucumber plants after the 7 days of seedling growth.
I'm Viva. Let me give a conclusion of what we have talking about our last few minutes. The PGPR colonizing the inner part of the roots play an important role in growth and development of the plants directly or indirectly. By treating the cucumber seeds with selected isolates significantly improve seedling emergence and growth. An important mechanism of plant growth promotion was IAA because it promotes the root development and uptake of nutrients in plants. By colonizing the PGPR on the root tips of plants, control the soil-borne diseases. In the present study, we have isolated 10 new strains of PGPR with several growth promoting traits such as antifungal activity and were found to be efficient in controlling Phytophthora crown rot in cucumber seedling. The PGPR strains are used for biofertilizer applications and as a commercial purpose biocontrol agents in the fields. Moving on to the next person.
Bibliography.
Acknowledgements. The authors would like to acknowledge the financial assistance from University Grant Commissions through Research Management Committee of Bangabandhu Sheikh Mujibur Rahman Agricultural University, Bangladesh.